Hey, what's up, everybody? Welcome to lab number 3 for AP number 1. This is our first lab where we're doing histology. Now, remember, histology is your study of tissues. And remember from lecture that a tissue is a group of cells with a common structure and a common function. Now, there are 4 basic kinds of tissues. There's epithelial, there's connective, and there's muscle, and there's nervous. Now what we're seeing here, really basically everything on this entire micrograph here is epithelial tissue. Notice all the nuclei, by the way, all these purple circles here are nuclei. So if I see a nucleus, I know I've got a cell. So here's a nucleus, and I can make out the outline of a cell around it. Okay, so epithelial tissues, what they do is they cover internal and external body surfaces. So the outside of your skin, that's epithelial tissue. The lining of the outside of organs, so the outside of your heart, the outside of your small intestine, epithelial tissue. The space inside the body, like this is a cross section of the trachea. The space in the middle of a tube-shaped organ is called the lumen, by the way. And epithelial tissue will line the lumen here. So any organ with an open space in the middle, so trachea, esophagus, stomach, bronchi, fallopian tubes, urinary bladder, all lined by epithelial tissue. So external and internal body surfaces, basically if it's exposed to a space, you're going to have an epithelial tissue there. Now, we actually can describe the 2 sides of an epithelial tissue. So here's some space outside the body. This is skin here. And this is all epithelial tissue. Like right here to here, this is the sort of more purplish is epithelial tissue. We see lots of nuclei in there. And we can describe this epithelial tissue in terms of 2 of its surfaces. The surface exposed to the space, whether that's the space outside the body or the space of a lumen, that surface is called the apical surface. Think about the word apical meaning or referring to apex, which is like the top of something, right? So that's the apical surface because it is next to the space. Here's that cross section of the trachea. Here's the lumen, the space. And this would be the apical surface, what I can just trace my cursor on. That's the apical surface there. Now, opposite of the apical surface. So this is all epithelial tissue right here. Apical surface is here. So the other side is called the basal surface, which is easy to see in this picture of the skin here. Harder to see in this trachea just because we haven't zoomed in enough. All right, so epithelia are always going to cover internal and external body surfaces. There's always going to be a space next to them. The part of the epithelium immediately abutting that space is the apical surface. Then the opposite side of the epithelium is the basal surface. All right, a couple other things about epithelial tissue. They are anchored to one another, the cells rather, are anchored to one another by these specialized connections. And there are 2 connections we've got to know. One are the desmosomes. So this is actually a super zoomed in close of the epithelium of the skin. Like this is the nucleus of a skin cell right here. This is the skin cell, the rest of it around it. Here's another skin cell right next door. And skin cells are attached to each other. And you can actually see all these little lines right here. And these things are known as desmosomes. Desmosomes. And what desmosomes do is they anchor cells together. So desmosomes are really strong. So desmosomes are going to anchor cells together, and they're going to do that with great strength. Think about how much damage and how much physical stress you put on your skin. You need skin cells to be attached to one another strongly. Desmosomes do that. Now, another specialized connection are the tight junctions. So this is actually the lining of the small intestine over here. So here's the lumen of the small intestine, the open space. This is the epithelial lining. I see a row of cells here. Of course, this would be an apical surface, and this would be a basal surface. And these individual cells here, so every time I see a nucleus, these are individual cells. It's kind of hard to tell where one cell ends and another begins, but I can kind of figure out it's like right there. And these two neighboring cells, they're attached to one another by these special connections called tight junctions. And what tight junctions do is they seal cells together. So basically they're going to tightly join cells, not so much with the purpose of strength, but with the purpose of not letting stuff get in between them. So remember, like I said, this is the lining of the small intestine over here. This is the space within the small intestine. And we don't want bad things. We don't want bad stuff to be able to sneak between cells, do we? No, not at all. We don't want to do that. So to prevent that, we're going to have these cells, these neighboring cells, really tightly affixed to one another with tight junctions, and it stops stuff from squeezing through. So these are two examples of specialized connections between epithelial cells. Now, I've got a bunch of little tubes in the kidney here. They were cut in cross section. So here is the lumen of a little kidney tube. Here are the cells lining it. This is the lumen, this is the space. So these are epithelial cells. This is an epithelial tissue. Here we put an arrow on the apical surface of those cells right there, and then I'll put another arrow on the basal surface of those cells, okay? And epithelial tissues are going to be supported by this thing called a basement membrane. So it's always attached to the basal surface. You can't always see it, but we can see it here. Do you see this pink line right here? Put some dots on this pink line. This pink line here, that is the basement membrane. It's like the supportive proteinaceous layer that's going to be attached to the basal surface of an epithelium. Now, you don't always see it. Like we see it nicely here in this particular tissue, but it just depends on your slide and your organ. You don't always see it, but it's always there. Okay, another fact about epithelial tissue. So we know they're exposed to a surface. We know they're the cells are going to be connected with specialized connections. They're also supported by tight junctions. Epithelia are also avascular. So there are no blood vessels within the tissue. There are going to be nearby blood vessels, but not any blood vessels in the tissue, which makes a lot of sense. Like, for example, if I go back to that skin picture here, this is the epithelium of my skin. Do I want blood vessels in it? No, because I damage the skin all the time. The epithelium gets broken all the time, but if I break my epithelium, I don't bleed, right? That's a good thing. Now, I will have blood vessels in the underlying tissue. I don't really see any at a quick glance here. I'll have blood vessels in the underlying and the neighboring tissue, but not in the epithelial tissue. So epithelia, in other words, are avascular. They lack blood vessels. Okay, we got some good basic epithelial characteristics. We also will describe epithelial tissue as being simple if they're composed of a single layer of cells. So if you just have one cell and then another cell and another cell and another cell, and they're just in a row like this, that is a simple epithelium. So no cells on top of each other, just one row. So like if you look at this tissue I have here, again, this is the lining of my small intestine. This is my lumen up here. And what I can basically do is I can look at all my nuclei. I can draw a line that goes through just about every single nucleus in this epithelial tissue. All right, just about every single one. There's a few outliers that just got squeezed out of place. But if I have one row of nuclei, I've got one row, one layer of cells, and it is a simple epithelium. Now, simple epithelia are not so great for protection, but they're good for exchange. Like it's good for, think about the small intestine. Nutrients need to get absorbed, through this layer. This is actually a blood vessel down here. This guy right here. I have a better circle around him. There he is. That blood vessel right there. I want nutrients to go through these cells and get in here. So, a simple single layer epithelium is good for that. All right. So good for exchange, good for moving stuff through. So as a simple epithelium. It's not the only possibility, right? Besides a single layer of cells, we can also have multiple layers of cells. So if we look at this picture here, this is my epithelial tissue from here up to here. And look, nucleus, 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 nucleus. So I got just cells stacked upon one another. Here's my lumen up here. And I got cells stacked upon one another. And if I have multiple layers of cells, that is said to be a stratified epithelia. This is actually the lining of the urinary bladder here. Stratified epithelium, of course, are not going to be good for exchange or moving stuff through them, but they are very good at protection. So what do you think your skin epithelium is made out of? Do you think it's simple or stratified? Well, of course, you're going to have to... Your skin, one of your jobs of your skin, there we go, is going to be protection, protecting you from physical damage, chemical damage, pathogens. So your skin epithelia will certainly be a stratified one. All right, so epithelia can be simple or stratified. We can also describe the shape of the apical cells. There are three standard shapes. One such shape is squamous, which is just another way of saying flat. So like if you look at this cell right here between the two arrows, this guy, he basically runs from here to here. This guy is very skinny, right? Very flat. That is a squamous cell. That is a squamous cell right there. Got more squamous cells. I got like a whole row of squamous cells here. All right, let's actually clear everything here. Like I'm putting a dot on all the nuclei of these squamous cells. Okay, the nucleus is flat. The cell is flat. Very good for exchange or for just not taking up a lot of space. And by the way, this is only one layer of flat cells. So you would actually call this epithelium simple squamous because it is a single layer, that's simple, of flat cells, that's squamous. Okay, another shape we've got is cuboidal. Cuboidal cells are roundish or squarish, and what I like to do is I like to look at the nuclei. Look at that nucleus right there, look at that nucleus, look at that nucleus. All these nuclei are basically circles. If the nuclei are circles, it is going to be cuboidal. And again, notice I have a ring of cells here. It is a single ring with round nuclei. So that is going to be a simple, because it's a single row, simple what? Simple cuboidal epithelium. Fantastic. So there's one final shape. The final shape is columnar, which is more of a rectangular-ish shape. Here's our, whoops, I didn't have my drawing tool going. There we go. Here's our lumen here. Here's our epithelium. Look at these nuclei. Look how oval they are. Look for oval nuclei. If you see oval nuclei, you know it is columnar. And by the way, here's apical side. Here's basal side. Blood cells in the blood vessel next door. So this is a single layer of oval rectangular cells. This is simple columnar epithelium. Okay, fantastic. Let's keep going. And so now we're going to put those words together, like those shapes and those layer types, and talk about epithelial tissues. This is simple squamous epithelial tissue. Simple squamous epithelial tissue. It's a single layer. The cells are flat. So this right here, I'm just going to draw a line on it. And I just draw one line on it because that's basically what it is. It's a line of cells. This is simple squamous epithelial tissue. This is actually in the lungs. And simple squamous epithelial tissue in the lungs is ideal because one layer, skinny flat cells, great for the exchange of gases. The lumen is just space for air. And in the lungs, the simple squamous epithelium, it forms these little pockets. This is a little pocket right here. A single little pocket like this is called an alveolus. The plural to that is alveoli. So lung simple squamous epithelium forms these little pockets, these little cavities, which are called alveoli. Okay? Good stuff. There we go. So here is some lung simple squamous epithelial tissue. Can you see the simple squamousness? Can you see the simple squamousness? Like, look at this. Look how just a skinny line, right? Lots of air space in the lumen. That's how you're going to know it's the lungs. Simple squamous. Little indentation right here. That is an alveolus right there. Little cavity right there. A little air pocket. That is an alveolus right there. Okay? So, that's some lung tissue. Excellent. Let's look at another example of simple squamous tissue. We actually looked at a very similar picture a moment ago. This is from the kidney. And in the kidney, you've got about, in each kidney, you got about 1 million, about 1 million of these things, these round things right here. This round thing is actually a ball of blood vessels. It's a ball of little capillaries. This ball of capillaries with the big G on it is a glomerulus. And you can see all the red blood cells inside it. So this glomerulus is where we filter our blood. All right, we filter our blood. It's the first step in making urine. And so we filter our blood in this glomerulus. Notice there is a space, there is a lumen next to it. And then adjacent to that lumen, adjacent to that space, where the red arrows are, there is an epithelium. And look, I can basically draw a line on it. If I'm drawing a line on it, it is going to be simple one layer, squamous flat, right? Simple squamous epithelium. This is actually called the glomerular capsule. This is the glomerular capsule where I'm again drawing that line. It's made of simple squamous epithelium. It's called a capsule because it encloses something, glomerular capsule, encloses the glomerulus. It creates this lumen. This is where I find filtered blood, which has a great name, filtrate. And filtrate eventually becomes urine. We do a few more things to it, eventually it becomes urine. All right, so that's some more simple squamous epithelium, just in the kidney this time. Here is another kidney. You're going to see this thing right here. What was this thing? It is a glomerulus. You're going to see this thing, and you're going to be like, that's the kidney, that's the glomerulus, and you're going to know next to it, right here, glomerular capsule, and you know that glomerular capsule, look at it. You can just draw a line on it, right? So it is simple squamous epithelium. All right, cool. Done with simple squamous epithelium. Let's move on to simple cuboidal epithelium. This is actually still the kidney. Still the kidney, just a different, we're zooming in somewhere else. We don't see a glomerulus. There's a glomerulus near here, don't worry. We just don't happen to have one in this picture. What I want to point out to you, though, is that I see a ring of cells. Remember, rings, here's another ring. Rings are created when we cut a tube transversely in cross-section. So I got lots of little kidney tubes here. I don't call them little kidney tubes. I call them renal tubules. Renal means kidney, tubules means little tubes. Now notice my renal tubule made of one row of cells, because I got one row of nuclei here, and those nuclei are circle-ish. They're circle-ish, right? So this is a simple cuboidal epithelium. Here's the lumen, here's a lumen, here's a lumen. And this simple cuboidal epithelium lines the lumen of the, and creates, in fact, the renal tubule. Now this tube would contain that liquid called filtrate, which is filtered blood. And what the filtrate is going to do, if there are nutrients, there are nutrients in the filtrate, and the nutrients are going to get absorbed. So if there's nutrients, the nutrients are going to follow the path of the red arrow here, and they're going to be absorbed into the cells, so you don't end up peeing them away. Any waste products are going to be added to the filtrate, so you do end up urinating those away. So this simple cuboidal epithelium here is really good at doing absorption and secretion. One cell layer is great for doing that, right? Because you don't have to go through multiple layers. A squamous one would not be as good, because squamous cells, by virtue of being so skinny, they can't have as much like cellular machinery inside them to facilitate the absorption and secretion. All right, by the way, notice we have this pinkish line here. We mentioned that a little while ago. Do you remember what that was? That was called the basement membrane. So we see this basement membrane here. Okay, good stuff. Good job. We're moving on. Got some more renal tubules here. Can you see them? Can you see all the lumens? So lumen, 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 two, and lots more. I'm just not doing all of them. Cells around the lumen, one row of nuclei, generally circular-ish nuclei, simple cuboidal epithelium. These are renal tubules. If ever I'm seeing little tubes in cross-section, I know they're renal tubules. Renal means in the kidneys. And I would have filtrate in these lumens. And these tubule cells are going to do absorption, secretion, helping turn that filtrate eventually into urine. Okay, cool. 2 more epithelia to do today, and then we're done for today, all right? Hopefully get out of here in about half an hour. Now, the small intestine. Here's the lumen of the small intestine. It is lined by a simple epithelium. Look, I got one row of nuclei. They're generally ovoid in shape, so it is simple columnar. Now, the lumen up here would contain something called chyme. Don't say chyme. When we have the pronunciation test, don't say chyme. It's chyme. Chyme is a mix of food, drink, and digestive juices. So the columnar cells here would absorb nutrients from the chyme. Take the nutrients in, add them to a neighboring blood vessel. Wastes would also be secreted into the lumen, so we could eventually poop those wastes away. So nice simple columnar epithelium here. Fantastico. Here I've got another view of it. So this is another small intestine here. Here's my lumen. Now in this case, the epithelium is going down this way, and we don't see it, but it's looping, U-turning, and coming back up this way. And so it has folds in other words. But again, it's basically a single row of nuclei, oval cells. So this is a simple columnar epithelium. And in this particular picture, we see some other cells besides the simple columnar guys. these guys, and I've got a couple labeled with the red arrows there. I'm just doing some more here. These are goblet cells. Goblet cells make mucus. The mucus is going to be lubricative. It's going to be a lubricating mucus to help chyme slide along. I just noticed this arrow over here. What is this arrow doing? I don't know. Let me know in class what that arrow is for. I was about to say, let me know in the comments, but I don't let people do comments on these. Anyway, good stuff. Let's keep on going. Here we see the same picture we just had. We just kind of it's just kind of cropped. Here's that simple cuboidal epithelium right here. Look at that single row of ovoid nuclei. Here is a goblet cell. There's another goblet cell. And then what I want you to notice is that in the lumen here, this is the apical surface next to it. And you can see there's something here. There's something like on these cells where I'm drawing a black line here. And that something are these cellular extensions. The cell membrane actually goes up and down and up and down and up and down. This is a super zoomed in version here. And these up and down extensions are called microvilli. Micro means little, villi means fingers. And microvilli give us more surface area on the apical side. And more surface area means I'm going to be better at absorption, better at secretion. All right. Good stuff, good stuff. Check out this one. I love this one. It's such a pretty picture here. And this is my lumen up top. Whoop, come on, drawbar. There we go. This is my lumen. Single row, ovoid nuclei, simple columnar. Now, this apical surface, what would you find here? You would find microvilli. Now, what's absent here? Just, they're just not in this particular stretch, but goblet cells. And of course, what do goblet cells do? Goblet cells make mucus. But look at this picture. This is an amazing picture. Lumen, single row of ovoid nuclei, simple columnar, microvilli, there they are. I don't see the individual extensions, but I see there's a border here, right? There's something going on here. And this clearish thing, that is a goblet cell, which of course makes mucus, which lubricates the lining of the intestine for easier slipping and sliding of the chyme. Okay, we got one more tissue type to do today. It is a weird one. Probably my favorite of the day, though. This is called pseudostratified columnar. Okay, check this out. Here's my lumen. This is actually of the trachea. The epithelium runs from here all the way down to here on the green bar right there. And what I want you to notice is here's a nucleus. Here's a nucleus. Here's a nucleus. Here's a nucleus. It's like I got nuclei stacked upon one another. It appears is the operative word there, appears that this is going to be a stratified epithelium, but it's actually not. It's actually simple. What's going on here is you've got one cell with its nucleus and then another cell next to it who's kind of leaning on it with his nucleus, and another guy who's leaning on him, and there's his nucleus, and then a really weird shaped guy. He's shaped like this, and there's his nucleus, but all of them are touching the same base here. They have the same basal surface. It's just the nuclei are all at different heights, even though they're all standing on the same surface. So they're actually simple, but they look stratified. So it's falsely stratified. In other words, pseudo-stratified. And the commonest shape is columnar. The standardist, commonest shape, not standardist, the commonest, not a word, I know that. The commonest shape is columnar. That's why we're pseudostratified columnar. Now there's also some beautiful goblet cells. Look at these nice abundant goblet cells in here. And these goblet cells make mucus, but the mucus in the trachea, which is what we have here, is different from that of the small intestine. Small intestine mucus, and we mentioned this a moment ago, small intestine mucus is slippery. It's slimy, okay, for lubrication. Trachea mucus, I'm just going to put some quote marks, is sticky. All right, why do you want sticky mucus in your airway? Well, think about it. When you breathe in so much junk, so much particulate matter. You breathe in so much dust particles. You breathe in bacteria, viruses, pollutants, pathogens, pollen, a lot of P words, right? And you want to trap that stuff. And that's what the mucus does. Now, the mucus doesn't do any killing in the trachea, but it does trap all the stuff, all the particles, pathogens, and pollen, all the P's there that you inhale. So your trachea has cilia. These hair-like things are cilia. They're like bigger versions of microvilli. Notice I can see them individually here. And what they do is they're constantly sweeping upward. They're doing a sweeping motion. Like imagine you're doing the wave, like you stand up and put your arms up in the air, right? So you're always sweeping mucus upward. You are constantly, unconsciously swallowing this mucus. It goes right up your trach and then directly down your esophagus. You don't even realize it. And this is good because then the acid in your stomach deals with all the trapped stuff. All right. I think we got one more slide to look at. Do you see these guys right here? When you see these guys, you're going to be like, cilia. When you see cilia, you're going to say to yourself, that's pseudostratified columnar, that's in the trachea. Boom, 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 done. And what do I have right here? And right here, I got some goblet cells, which of course make mucus. What kind of mucus? A skinny mucus. And that, my friends, is it for lab number 3. A little more than half an hour, but not bad. Thanks for staying. If you have any questions, come see me. Come visit, talk to me. All right, till next time. Bye-bye.